Hi folks, welcome back to our last episode of the Visual System series. Today we're diving into some mind-bending optical illusions and discussing a famous face you might know, that is Jennifer Aniston. So grab your popcorn and let's get started. After a very thorough exploration of the primary visual cortex, it's time to move forward and see what's coming up next. V1 sends strong feed-forward connections to the secondary visual cortex, also called V2. And here is a disclaimer. That's the case for animals with rather complex visual systems like humans, other primates, but also cats, ferrets, or tree shrews. In mice, for example, there is no distinct secondary visual cortex. In mice beyond the primary visual cortex, we talk about higher visual areas in general. These areas are a part of what is commonly called the association cortex. The organization of higher visual areas in mice is still an area of active research, and while there is a lot of similarities, we need to keep in mind that the mouse cortex has 8 times fewer neurons than the human cortex. Therefore, it's much simpler and not every human brain region has its equivalent in mice. Moving from the primary to the secondary visual cortex, we notice a key feature. The retinotopic map flips. Scientists can determine this flip using imaging techniques to map retinotopy. And while the secondary visual cortex may not always mirror the V1, this flip serves as a common rule in identifying higher visual areas. Let's talk about the anatomy. Each hemisphere houses its own V2. It's composed of the dorsal and the ventral parts. Just like V1 and the rest of the cerebral cortex, V2 is composed of 6 layers. And neurons in V2 share many properties with those in V1, like tuning to specific orientations, directions, or spatial frequency, etc. But they also have new, more complex properties. That's when things get funky and our brain can trick us into many optical illusions. Intermediate level visual processing is like putting together puzzle pieces to see the bigger picture. Your brain is really good at guessing how things fit together, even when the information it gets is a bit fuzzy. It's a useful property of our brain, but sometimes these guesses can lead to seeing things that aren't really there, like the edges of a shape in an optical illusion. One famous example is the Canissa triangle illusion, where our brain perceives a triangle that isn't explicitly drawn, but is created by the illusory contours formed by 3 Pac-Man figures. It's a cool trick our brain plays on us as it tries to make sense of what we see. Another really cool feature of the secondary visual cortex is that it figures out the depth and helps us tell objects apart from the background. How does it work? Well, both eyes see slightly different images because they are set apart. That's what's called the binocular disparity. By the way, binocular disparity is also used in virtual reality goggles to create a sense of depth. Now, neurons in V1 and V2 use these differences to figure out how far things are. When an object is closer, the images from each eye are more different, and when it's further, they are more similar. The neurons analyze these differences to create a 3D reconstruction, helping you see depth and tell objects apart from the background. Some neurons in V2 also have a unique feature called border ownership. They get active only when an object, but not the background, is to one side of the edge. And that's when another optical illusion can kick in. You might be familiar with this image. It's a famous example of a bistable figure where we perceive either 2 face profiles or a vase at a time, but not both simultaneously. V2 uses cues like which object surrounds another to establish depth and relationships. But as the higher levels of visual processing come into play and try to identify the objects as a whole, contradictions creep in. The brain, at this more advanced stage of interpretation, is trying to shape the overall perception and struggles to decide whether the vase or the faces should be considered the main focus. That's when our interpretation of the image can flip between the 2 perceptions. Now it's time to mention an important concept, which is the 2-stream hypothesis. It's a model where visual information undergoes simultaneous processing in the ventral and dorsal streams as it exits the occipital lobe. The ventral stream, known as the what pathway, specializes in object recognition, while the dorsal stream, the where or how pathway, focuses on an object's spatial location relative to the viewer. This model, widely acknowledged in textbooks, serves as a guiding framework for researchers studying the visual system, but we need to remember that different brain regions don't work in isolation, but they are strongly interconnected. So despite being distinct pathways, these streams functionally connect and form a dynamic network that adapts its communication based on the specific task. Now let's move on to the next level and talk about the visual area V3. It consists of two functional subdivisions, the dorsal and the ventral part. The dorsal segment of V3 is conventionally associated with the dorsal stream, receiving inputs from V2 and V1 and projecting to the posterior parietal cortex. Neurons in this region exhibit responses to the coherent motion of large patterns. In contrast, the ventral V3 exhibits notably weaker connections with V1 and stronger connections with the inferior temporal cortex. Finally, let's dive into area V4, a segment of the ventral stream. V4 is a hub that combines details about color, brightness, texture, and the shape of objects. Within V4, there are modules specialized in sensing color. Neurons in V4 are sensitive not only to orientation and spatial frequency, but also to more complex shape properties. Extensively explored in the realm of visual attention, V4 excels in detecting important stimuli, leading to increased neuronal firing in response to these stimuli. But also, V4 plays a significant role in process of object recognition. The V4 visual area is key to our color perception, particularly in the concept of color constancy. Color constancy is the idea that we see objects maintaining the same color despite changes in lighting conditions. For instance, a red apple looks red whether it's in a shadow or under sunlight, while some other aspects of color constancy may initiate in earlier visual areas like V1 or V2. V4 stands out for its significant role in this process. Studies using specific stimuli revealed that changing the background illumination influenced the response of V4 neurons, aligning with lighting changes and corresponding to how we perceive colors. Here's an excellent example of how this mechanism can trick us into another kind of illusion. You can see a cube, similar to a Rubik's Cube. There is a square right in the middle of the cube on its side that looks orange. Now, if you take a look at the square in the middle, on the top of the cube, it might seem brown. But here's a surprising fact. If you take these two squares and look at them separately, they are actually the same color. So while the colors are truly the same, our brain adjusts the way we perceive them based on the lighting conditions. This is a super simple example on how our perception depends on the context. Next, let's dive into the final stage of the ventral stream, the inferotemporal cortex. Ever heard of it? Maybe not, but you might find it interesting that this area is home for what's called Jennifer Aniston cells. Curious? I'll clear it up. Depending on who you ask, this region can be split into the occipital and inferotemporal cortices, or just grouped as the inferotemporal cortex. This is where the magic of higher visual processing happens. It's not just about receiving signals from the eyes. This area integrates both sensory data coming from earlier visual areas and the top-down input like context and semantic significance to make sense of what we see. If the term semantic sounds a bit abstract, let's use a simple example. Imagine you're in a park and you see and hear a bird. You could just experience this moment, seeing the colors, hearing the chirps, without attaching any labels or names to what you're observing. This is focusing solely on the sensory signals. On the other hand, you could recognize and name what's around you. That's a bird, that's a tree, that's a pond. This is focusing on semantic signals, where you're adding meaning and significance to what you see and hear. Hopefully this makes this idea of semantic more tangible and easier to understand. In the inferotemporal cortex, or IT, the cells are organized into columns that each respond to similar types of visual information. These columns extend through the entire thickness of the cortex, allowing for different stimuli with common features to be processed in overlapping areas. At this advanced level of visual processing, our perception of objects is not just about what we see. It also heavily relies on our past experiences, memories, and other senses, as well as our emotions and intentions. This means that object perception at this stage is a blend of visual input and cognitive processing, creating a rich understanding of our surroundings. One interesting approach to understanding the role of a brain region is to look at the consequences when it's damaged. For instance, lesions in the temporal lobe of primates lead to a condition known as the visual agnosia, where there is a loss of the ability to recognize objects. In humans, visual agnosia comes in two main forms: aperceptive and associative. Aperceptive agnosia affects the ability to perceive or copy complex shapes or objects. This happens because the process of integrating visual features into a coherent object representation is disrupted. On the other hand, people with associative agnosia can still match or copy objects but struggle to recognize what they are. This is due to a breakdown in linking what they see to their knowledge of the object's meaning or function. Typically, aperceptive agnosia is seen with damage to the posterior part of the inferotemporal cortex, while associative agnosia occurs with damage to the anterior part. Within the inferotemporal cortex, certain neurons are dedicated to encoding highly significant objects like faces and hands. Some neurons are particularly activated by specific views of faces, such as the front or side, and others by facial expressions. An extreme case related to face recognition is prosopagnosia, or the inability to recognize faces, often caused by damage to the small area within the temporal lobe. Individuals with prosopagnosia can describe a face, recognize expressions, and identify it as a face, but cannot tell whose face it is. Interestingly, neurons that are highly responsive to faces are not scattered randomly, but are clustered together in densely packed groups. Ever heard about grandmother cells? This term refers to a neuron that's thought to be activated in response to a specific, complex, and meaningful stimulus, like the image or concept of your grandmother. Originally proposed by Jerry Lettvin in the late 60s, the idea was popularized in the media around 2005 after researchers at Caltech and UCLA found neurons that respond exclusively to images of celebrities like Jennifer Aniston or Halle Berry. The term grandmother cell originally described a cell that could respond to various modalities but has been predominantly associated with visual recognition. In a groundbreaking study, scientists used electrophysiological recordings in patients with epilepsy to show that some neurons fired in response to different pictures of the same person or even their name, but not to other stimuli. For instance, one neuron activated for various images of Jennifer Aniston, while another responded to images or the name of Halle Berry. The media often oversimplified these findings, suggesting that a single neuron could be solely responsible for recognizing a person. However, the reality is much more complex. These neurons, while they do respond to specific individuals, are a part of a larger network. They receive inputs from thousands, perhaps tens of thousands of other neurons, both bottom-up and top-down. It's as though these neurons sit at the pinnacle of a mountain of associated neurons, each contributing small pieces of information that culminate in the recognition of an individual. To be able to recognize objects, we also need to generalize to different conditions, and that is called perceptual constancy. It means that no matter if you change the size, position, or rotate the object, we still perceive it as the same object even though it creates a different image on the retina. It has been proven that lesions in the IT lead to failure of size constancy in monkeys. While in intact IT, the neurons respond constantly and invariantly to the same object regardless of their size. Another, and my personal favorite type of constancy, is visual categorization. That's the perception of individual objects as belonging to the same semantic category. I love this topic because this is the focus of my PhD project, but in this video I'll restrict myself to talking only about the essentials. When categorizing, we group objects belonging to the same category, but also discriminate objects of different categories. So if we look at apples, they don't look identical, and yet we can label all of them as apples. But when we take something else, we know that it clearly does not belong to the apples category. By creating categories, we simplify our behavior and the brain's computational load. We don't have to relearn what an apple is every single time we see one. Interestingly, there are category selective neurons in the brain that respond specifically to objects within a certain category, but not to those outside of it. Pioneering studies by Earl Miller's group in the early 2000s laid much of the groundwork for our understanding of this process. Our lab is exploring these concepts further in mice, and it's really exciting stuff. Trust me, it's not just because it's my work. These findings help illustrate how different brain regions like the inferotemporal cortex and many others are involved in visual associative memories. During the learning process, the synaptic connections in these regions change, which is a key to how we memorize and create associations. Okay, now let's explore the dorsal stream of the visual pathway. This pathway stretches from the primary visual cortex to various regions in the parietal and frontal cortices, including the middle temporal area and the frontal eye fields. Unlike the primary visual cortex, which organizes space into a retinotopic map, the parietal cortex divides its spatial representation into 4 distinct areas, each tailored for different motor systems. These areas are directly linked to specific movement control regions in the premotor cortex. For instance, the medial intraparietal cortex, MIP, targets arm movements and connects to the dorsal premotor area, while the lateral intraparietal cortex, LIP, is involved in eye movement targets projecting to the frontal eye fields. The anterior interparietal cortex, AIP, focuses on grasping and extends to the hand control area, and the ventral intraparietal cortex, VIP, is linked to facial stimuli and connects to the face control area. The dorsal parts of the visual system play a huge role when we're getting ready to interact with stuff. So they communicate heavily with the supplementary area and the premotor cortex. Take lifting a box, for example. You see a big bulky box and your brain automatically thinks, this is gonna be heavy, right? So you brace yourself to lift with a lot of effort. But when it turns out that it's super light, it gets very confusing and you might look funny. Parts of the visual imagery are constructed in higher resolution by eye saccades, focusing the fovea on important parts and therefore creating selective visual attention. When you look at this video, your attention selects specific objects for further visual examination. Maybe right now you look at me when I talk, or maybe you just dozed off and look at the objects surrounding me. We divide this attention into voluntary and involuntary. You looking at me when talking is an example of voluntary attention. But if something randomly suddenly appears in the scene, your attention would be probably pulled away involuntarily. A clear demonstration of how vision and attention are connected can be seen in patients with unilateral visual neglect, often caused by lesions in the posterior parietal cortex, PPC. These patients may struggle with tasks like copying drawings, particularly to the side opposite of the lesioned hemisphere. For instance, when asked to draw a clock, a patient with a lesion in the right hemisphere might omit the numbers on the left or squeeze them into the right half. Now think about this. Our eyes move around all the time, but why does the word not blur as our eyes make these rapid movements? This stability is partially due to saccadic suppression, a process where the brain temporarily reduces vision perception during rapid eye movements to prevent a blurry, jumpy visual experience. That's why our vision is not as shaky as the homemade video with a handheld camera. Before a saccade, neurons in the parietal cortex adjust their sensitivity, shifting focus to anticipate the post-movement visual field. This neural adjustment helps maintain a consistent perception of the environment during the quick eye movements. Mechanisms like visual masking and corollary discharge may play critical roles in saccadic suppression. Visual masking allows high contrast images to dominate over low contrast ones seen in quick succession, enhancing visual clarity. And corollary discharge from a posterior colliculus to the frontal eye fields signals the brain to ignore transient visual disruptions during eye movements, ensuring a stable visual experience. It's like a courtesy heads up from the superior colliculus to hold off on processing visual scenes as the eyes are rapidly moving. We've covered a lot of ground in today's episode, diving deeper into how our brain interprets the world around us. We explored the transition from the primary visual cortex, or V1, to higher visual areas like V2, V3, and V4. We looked at how optical illusions play tricks on our brain, using simple visual cues to create shapes and edges that don't actually exist. We also learned about depth perception and how our brain uses binocular disparity to understand the distance between objects and the background. We touched on the two streams hypothesis, where the ventral stream helps us recognize objects and the dorsal stream tracks their spatial location. Then we went even deeper into face and object recognition, discussing how the inferotemporal cortex helps us identify complex objects like faces and hands, sometimes with specific neurons like Jennifer Aniston cells responding to familiar faces. We also explored visual constancy, which allows us to perceive objects as being the same no matter their size, lighting, or angle. And finally, we talked about visual attention and stable vision during eye movements. All right, we made it to the end. Congrats on finishing the whole series with me. I hope you found something interesting for yourself. Let me know what you think and what else would you like to hear about. I hope to see you soon. Thanks.